I'm actually giving, as Tony said, 2 talks. Peter Ryan couldn't be here today, but I'm involved in that project, so I know it imminently. So I want everyone to cross their fingers, one hand, cross their fingers very hard to keep the video clip gremlin away, okay? Because I am relying on that. Anyway, so my sort of talk is about mosquito control. We still need to kill them and count them, because some of the new methods don't, you'll see, don't necessarily sound like we need to kill mosquitoes at all. So dengue fever is typically known with rash, severe retroorbital headache, muscle ache, vomiting, etc., but it's not fatal. Anyone in here, have they had dengue? No, oh, one person here, excellent. It wasn't any fun though, was it? Oh, okay. You can tell we're in Canberra. There's no tropical diseases here. However, there is a fatal form, dengue hemorrhagic fever, that rarely occurs. It's not that common. It involves plasma leakage. So you leak the plasma out, then you go into shock. And it only occurs in at most about 1% of dengue cases. And of those cases, 1 to 5% are fatal, although with modern care facility that knows how to treat dengue, that's usually well below that amount. And globally now, there's about 100 million dengue cases a year estimated, and about 40,000 deaths. So not a big killer, but it has a huge burden. The tallies are high because you end up with so many people coming down. 50% of the community can come down with dengue in a big outbreak. And there is no licensed vaccine. There are vaccines being tested, being developed, but nothing licensed yet. And don't worry about the details here. The main thing I wanted to emphasize here on the top, some people might recognize that young man on the left. Professor Richard Russell used to be at University of Sydney. That was him in his Billy Connolly period. But let's say Richard's gone to Bali, as Australians do, and gets bit by Aedes aegypti that's carrying dengue virus. And approximately 8 to 10 days later, that mosquito is infective, can actually transmit the virus to, say, Mike Lindsay over here from Perth. And he could get infected. And then he'll transmit the mosquito to local Aedes aegypti. So this is what happens in Cairns. And as Nigel said, it's a pan-tropical disease. In fact, you could argue that this is areas that are receptive potentially to dengue. It's not all areas where they have it. But the idea to show you there is that there's a big swath of tropical and subtropical areas that are at risk of dengue transmission. And globally, I call dengue the other pandemic. And dengue has been increasing, without a doubt, within the last 4 or 5 decades. Mainly urbanization, a lot of plastic, so containers that go out, people's yard, rubbish, fill up, breed mosquitoes, and a lot of international air travel to move the virus around. And Australia, we have the same thing. So this is viremic imports into North Queensland. And you can see that's been going up. It used to be steady around 10 a year. In the last few years, it's gone up to, well, we went over 50 in 2010. And we were getting about 30 cases a year. These are the ones that we know about. There's other ones coming we don't know about. And dengue outbreaks in North Queensland by month. And I guess the take-home message there is that we tend to get some big outbreaks and then have a few quiet years. And also, 2009 was a real biggie. And we had an article come out in PLOS One. I invite you to look at it because it's really interesting. And one of the features of that outbreak was the virus was going through the mosquitoes quicker than normal, and therefore our mosquito control was later than normal, finding out about dengue and being able to successfully intervene. Okay, so the mosquito carries it, Aedes aegypti. I call it the cockroach mosquitoes. So it's domestic, it lives around the house, it feeds almost exclusively on humans, very intermittent feeding. So one mosquito might feed on half a dozen people in order to get a full blood meal, because it's very good at avoiding the slap. And finally, this leads to explosive urban epidemics. So the mosquito likes hanging out. It loves old Queenslander houses. Dark, shady areas under the house, inside areas up there, the laundry area, wardrobes under beds, dark nooks and crannies inside the house. There is a dengue fever management plan, and it involves run by Queensland Health, and it's disease surveillance, mosquito surveillance control, public education, blah, blah, blah. But the bottom line for control strategies, we're still using pretty old technology. We have to send guys inside houses to spray residual synthetic pyrethroid insecticides. So it's guerrilla warfare, house to house guerrilla warfare against the mosquito. It works, but it's laborious. So one of the new things is Scott O'Neill from Monash University, and he's got this whole Eliminate Dengue team. And I'm going to tell you now, with little luck, the story, or someone's going to tell you the story of how Wolbachia could displace or replace this guerrilla warfare pesticide approach to controlling dengue. Okay, baby, let's go. Let's see. No, that's not it. Oh, here we go.
Dengue is a human virus, but the mosquito, Aedes aegypti, is responsible for transmitting the virus between people. As recently as 400 years ago, the mosquito was only found in Africa. Today, Aedes aegypti infests all tropical regions of the world, and with no specific treatments or successful control method, is causing enormous damage through dengue transmission. Our research is concentrating on a natural bacterium called Wolbachia that lives only within insect cells, but is passed from one generation to the next through the insect's eggs. Estimates are that there may be up to 5 million different species of insects on the planet, and that up to 70% of those species are infected naturally with Wolbachia. Insects that include butterflies, moths, fruit flies, and dragonflies, and mosquitoes that bite people, but not mosquitoes involved in the transmission of diseases such as dengue and malaria. Wolbachia is so common in nature because of the influence it has when insects with and without Wolbachia mate. If you imagine a female insect and a male insect, and the male insect has Wolbachia and breeds with a female that doesn't, then the eggs she lays won't hatch. But if you then imagine the opposite, when the female has Wolbachia and the male doesn't, she would lay her normal numbers of eggs, and they would all hatch, and they would all carry Wolbachia. You could also imagine a third cross, and that would be both parents carrying Wolbachia. The outcome of that cross would be, again, all individuals that were produced would carry Wolbachia. So, very quickly, over a few generations, the number of individuals carrying Wolbachia increases rapidly until nearly all the insects within a population have the bacterium. Scientists have been working on the biology of Wolbachia for a long time. In the 90s, Seymour Benz's lab in the US found a strain of Wolbachia in fruit flies that roughly halved the adult lifespan of the fly. Mosquitoes live to about the same age as fruit flies, around 30 days. But what's really interesting about dengue is that it takes 8 to 10 days after a mosquito has bitten a person with dengue before it can pass on the virus to another person. And what that means is the mosquitoes that transmit dengue are all old. So we had an idea, and that was, what if we could take the bacteria that shortens the lifespan of the fruit fly and transfer it to the mosquito that transmits dengue? And maybe it would shorten its lifespan, and, who knows, possibly reduce its ability to transmit dengue. That was the idea, but could we do it? We knew it wasn't going to be easy to move bacteria between species. We wanted to take the Wolbachia from the fruit fly and put it into the mosquito, and the only way we thought we might be able to do this was through microinjection, using a very fine needle and injecting Wolbachia directly into the mosquito's eggs. It took us thousands and thousands of attempts over many years, but we finally did it. We had a mosquito that carried the life-shortening strain of Wolbachia. The next question was, would it now do what we hoped it would, and shorten the lifespan of the mosquito? And yes, it did, but it didn't just do that. After testing the life shortening, we tried another experiment, and something happened that we really didn't expect. We gave the mosquito with Wolbachia dengue. The Wolbachia acted like a vaccine for the mosquito. It was blocking the virus from growing. And if the virus can't grow in the mosquito, then it can't be transmitted to another person. This was an even more exciting result for us than the life shortening. Our next step was to see if we could get the Wolbachia to spread from our mosquitoes into wild mosquito populations. We conducted months of experiments in large outdoor cages that we built in northern Australia.
Cans.
We filled the cages with mosquitoes that didn't have Wolbachia, and then introduced a small number of mosquitoes that did have Wolbachia. It didn't take many generations before all the mosquitoes had Wolbachia. While we were carrying out the experiments in the cages, we were talking to communities in Australia about the next stage of our work. With community support and government approval, we then began a series of releases of Wolbachia mosquitoes in 2 Australian locations. Our hope was that our mosquitoes would breed with the wild-type mosquitoes in the area, and so introduce Wolbachia into local Aedes aegypti mosquito populations. And we were successful. Those communities still have Wolbachia-infected mosquitoes. Now, our idea has developed to thinking about spreading Wolbachia through mosquito populations in countries where dengue is found. It is our hope that we may have some impact on reducing the estimated 50 million cases of dengue that occur each year around the world.
So remember, you say, oh, you don't have to kill mosquitoes. You don't have to catch mosquitoes. Oh, we see why you got to catch it for Wolbachia. So it's the idea, all paradigm of mosquito control is gone. And I used to work for this mob in Florida, and they used to kill mosquitoes in a big way. Of course, now we're releasing mosquitoes. And of course, we also need to be able to catch and count. This is actually a picture of Peter Ryan, who should have been giving that talk earlier. So, if we're going to put Wolbachia in, we've got to know catch mosquitoes to show it works. Again, remember the old Queenslander? Mosquitoes hiding in the Queenslander. We do have an exciting new pesticide here that works, and it could be what I call the barbecue reviver, this thing. Let's see if we can get this one going. Okay, you can put it in now, Chris. This is called methylfluthrin. And it's a vapor-active synthetic pyrethrin. Put the lid on. Great. That's good. Don't worry, cameraman gets his act together in a minute. But this is just a little strip, little plastic strip. That'd be heavily overdosed. But you can imagine putting this into some sort of room. And it's 30 seconds. Okay, I think that might. Last man standing falls down at a minute. Oh. Okay, so we don't have to worry about this. So we did a methylfluthrin trial some years ago, and this is published in American Journal of Tropical Medicine and Hygiene. A Tasmanian called Luke Rapley did this work as a postdoc. And basically, we had a couple of rooms that went together, and they were connected by a door here, an open door, and we marked these mosquitoes different colors so that we could tell what room they came from. And I'm just going to cut the chase, but here was a methylfluthrin device in one room, the other room had nothing. And I don't have time to go into a lot of details, but basically, when we use methylfluthrin versus a standard mosquito coil and a control, we killed mosquitoes in both rooms. And the mosquitoes from the untreated room actually flew into the metafluorin room. So and here's one of the new emanators. That's that thing you called Sumi mesh. And it's a 5% mesh, although now they moved to a 10% mesh. One of the problems with that room trial was that the room had screened windows. So I thought, well, what would happen if they could fly out the window? Then you just handball the dengue-infected mosquito to your neighbor. So I thought, we've got to do a room that has open windows. And so this is an old bedroom in my old Queenslander. I've got a sign on the door, do not enter, mosquito experiment in progress. My wife don't dare open that door. Anyway, so we had 2 of these windows like this that were wide open, and we released cohorts of mosquitoes in here, opened the metafluorin device, had a fan going on low, and here's the results that we got. This is at 1 and 2 devices. And you can say, why 2 devices? That's cheating. But no, they've gone to the 10% metafluorin device, which is the same as 2 5% devices. The bottom line is that we would release 10 mosquitoes in a room, and most of them didn't fly out the window. Most of them died within a couple of hours. And the number that escaped was pretty low, like 1 or 2. So that's pretty cool. The window's wide open. They could fly right out the window. The other thing I did, and this seems crazy because my eyes aren't all that good, but the room was white. I sat and observed individual mosquitoes. I'd let them loose, open the device, and see what happened to them. And here, so blue is down here, is biting mosquitoes trying to bite me. Then is flying around the room. Then is sitting on the suitcases I had in the room. And finally, the purple up here is knocked down when mosquitoes fell down and died. And that's what happens. And you can see that the biting quit within a couple of minutes, and then the mosquitoes were agitated, like you saw in that video clip, flying around, and then they just sort of went to bed, more or less, and then they just fell down. Now, the windows were wide open in this place. It's fascinating. I saw one mosquito fly out the window, turn around, and come back in. Go figure. But they're endophilic. They love being in the house. They can't help themselves. So that is an exciting pesticide. You'd be able to hand out a dengue outbreak. Maybe you give the people like 5 or 6 of these things. Say, put them in the rooms that mosquitoes are biting. Maybe you're going to have that barbecue. You tell Nigel, Kai, I got metafluorin. The Barbie's on, mate. No, it'll work. I hang it right down by your feet. It works, man. It works. You're not getting it in my house. So. Okay, we still need to count the mosquitoes, too. You've seen the Wolbachia stuff. You look at the very early Nature paper, and they used ovid traps. Ovid traps are basically a water bucket with a paint paddle or a strip of cloth in there, and the mosquitoes lay their eggs on them. Pain in the butt, because you can't just count the eggs. It could be, as Nigel found, up to 17 species. So you've got to be able to rear these things out. That takes time. We've also used sticky ovid traps, and sticky ovid traps they're reasonably good, but they're catching mosquitoes. But look, they're messy. That glue is messy. So one of the things we did was we developed a concept, what I call a fly to the light concept of mosquitoes. And we've all remember when we were kids, at least I do, we used to catch like bugs in a jar, bees in a jar and stuff like that. And you notice that the bee, or here in this case, a monarch butterfly or a wanderer, they fly around the sides of the jar trying to get out through the clear areas. Positive phototactic. You could literally take that jar, turn it upside down, pull the cork out, and a mosquito wouldn't find its way out. And we developed this. There's a guy in this room, I'm not going to point this gentleman out, named Giles Cordes, and he developed this trap in consultation with me, where mosquitoes are attracted. This goes to a CO2 regulator, and the CO2 comes out of this pipe. That's how mosquitoes find you, quit breathing, and you won't get bit. And the mosquitoes follow the CO2 in, and then they're inside this sort of translucent chamber, and they don't find their way out. So we thought, how can we modify this? Here's an example, one of those traps. There's no lights, there's no fan, there's no power. It's what we call a passive box trap. So it really does catch mozzies. So how can we modify this to make it work for Aedes aegypti? And a lot of the Aedes aegypti traps are like sticky traps that have water at the base, and the mosquito comes in and gets caught on glue, like that glue trap we saw. So we developed a couple of methods where we use the flight of light concept. The mosquitoes come in, and then this is translucent, and we treat the inside with pesticide. And they come in and they try to get out, and they can't find their way out through the little opening on top, and they die. And so here is some data from a double sticky over trap, a traditional method, and here is what we call the gravid Aedes trap. And you can see that not only have we replaced the glue, but we got a trap that actually catches more mosquitoes than the sticky trap. So we're quite happy with that. We also compared it to the BG trap. Here's the BG trap that we've seen. And this is sort of the industry standard for Albopictus and Aegypti. And they're attracted to the black. Mosquitoes like black. Black means shade. Shade means no wind. Shade means high humidity. And it's just like an Aussie on the beach. Where do you go? You find them in the shade under the almond tree. And so the mosquitoes are attracted to this and get sucked down by the fan. This is what they're using in the Wolbachia program right now. One issue, two issues. One is these things are about $200 a pop. Secondly, they require power. So you can see the power line it's plugged in, main power. So if you're going to Vietnam, you're going to Indonesia, you're going to favelas in Brazil, they may not have access to power. So the mandate was try to find something new. So we did compare the BG. We had two trials where we compared the BG and the GAP. And I think this is the first trial we ever did. It's a bit of an Ashton-Agar moment where you probably do better than you probably should. And you can see that we caught almost as many as the BG. And then we did these swap trials and we got about 70%. But I reckon it catches about 50% or so as many females as the BG trap. And finally, I'm going to mention some work with Ari Hoffman at the University of Melbourne. So this is the mosquito populations of popcorn, Aedes aegypti in Babinda. We were doing the releases then, so the numbers went up. The thing you'll notice here, remember popcorn's not real fit, it's life-shortening, the eggs die in a month. The populations almost went down to nothing. So the concept would be, if you had an isolated community and wanted to get rid of Aedes aegypti, what you might be able to do is get popcorn in, and then it'll self-destruct. It'll self-eliminate. And as Nigel showed you, there's a lot of these little towns out here that are very close, that have aegypti very close to Brisbane. So we'd be interested in trying it. So that might be a powerful way forward where right now there are other groups using GMO methods to tackle that particular problem. And finally, I've got a few people to acknowledge. All the names are up here, but the Eliminate Dengue team, Ari Hoffman, my staff at JCU, Greg Devine at QAMR now, and Richard Russell and Joe Davis. And finally, I'll leave you this slide. If we don't make these new technologies work, then we're left with some pretty old techniques. And Hare is the guy who first described dengue hemorrhagic fever down here. And that's the kind of stuff they used to do.